Good afternoon, everybody. My name, as Jake said, is Axel Imhof. I'm a professor at the University of Munich, and I'm the last addition to the Scientific Advisory Board of Volition. And what I'm going to do for the next 10 minutes or so is to tell you why I'm so excited about the Nu.Q capture assay and why I think that could really serve as an enabling technology for Volition to further improve the Nu.Q technology. As Jake mentioned, in ELISA assays, as the Nu.Q is based on, as the Nu.Q test is based on, what you do is actually you capture nucleosomes at the bottom of a plate out of plasma or serum, and then you detect one particular feature with another antibody that is actually labeled. And if you get label bound, then you get a signal. And if that feature that you're looking at is something that's specific for a tumor, then the signal that you get is proportional to the amount of tumor cells that have lysed in the body. Right? However, this is one feature that you can look at, and of course, wouldn't it be nice to look at more features and to have more features, to have panels, so that would allow you to discriminate between different tumors and different tumor stages, and this is where the Nu.Q capture idea is actually coming from. So what we're doing is we're actually taking a similar strategy where we fish out the nucleosomes, but then rather than taking one antibody and looking at one feature, we actually try to look at multiple features, ideally looking at all the features that are possible in the nucleus that we fish out. And of course, I brought you here a 10 million increased model of a nucleosome. So this is how a nucleosome looks like. And what you immediately can see, there has two parts. It has DNA, that's the red bit, and it has proteins in the middle. So you can look at both features. Of course, the DNA, you can look at the DNA sequence and determine the sequence that you fish out. And the, in my opinion, more relative and more relevant and more epigenetic part is to actually look at the white bit, which are the proteins that make up the nucleosomes. And if you have some time later on during fish, you can actually look at this, and what you'll see immediately is that there are clearly more than one feature. And in fact, by using mass spectrometry, and this is what we've been doing for the last 20 years or so, you can detect up to 200 different features on the nucleosome in a or on the histone in a highly parallelized manner. So rather than in ELISA, where you actually look at one, you can look at those 200 features from one sample. The disadvantage is that you need more sample, and this is why we actually established this method where we moved away from the ELISA plate and used beads to fish out the nucleosomes. By that way, we can take larger amounts of plasma or serum and purify the nucleosomes from. And so in preliminary experiments, what we just simply did, we took recombinant nucleosomes and just checked whether we can pull down those nucleosomes from a mixture and whether we are able to deplete this. And in fact, we can do this. So, sorry. Oops, no, it was too fast. Which you see here on the green bars, we add synthetic nucleosomes and we look at what is actually remaining in the supernatant, and you see that we can add quite a bit of nucleosomes and fish them almost all out and deplete it. That does not only work with synthetic nucleosomes, it also works with plasma, where we can fish out most of the nucleosomes out of plasma. And that, of course, now allows us to do nucleosomics, or to actually use those nucleosomes to detect features and learn something about the nucleosomes that we fish out of the plasma. And we can do this by DNA sequencing, looking at the circulating tumor DNA, and we can do this, and this is where my expertise comes in, using the histones and using mass spectrometry. And so the preliminary experiments told us that we were able to immunoprecipitate the circulating nucleosomes from plasma and serum, and we're now able to extract the DNA, and the goal is ultimately for the sequencing to look at the circulating tumor DNA. Now, what we have done is we actually separated the DNA from the proteins and looked at the proteins and did mass spectrometry in this. Indeed, we could already find more than just one epigenetic feature in those circulating nucleosomes, multiple modifications, multiple variants that are all potentially indicative for different types of tumors. So just to summarize on the Nu.Q technology, we're actually able, and that's what we can show in those preliminary experiments, to pull out nucleosomes from a highly complex mixture such as plasma or serum up to a depletion of 90%. The next step is, of course, now to go into tumor samples to see whether for particular tumors we see particular features, and then ultimately to allow Volition to actually develop antibodies that can then be used in the ELISA platform, ideally in a pattern so that you can actually really distinguish different types of tumors. So I'll just skip this here. The mass spectrometry is clearly an enabling technology in a shotgun way where we can go one step ahead and then try to do some sort of biomarker discovery where on the nucleosomes that serve as biomarkers clearly for tumor, we can detect different features, and those features on the nucleosome will then allow us or allow Volition to develop specific antibodies that one can use in multiple assays and then further and much more precisely do the diagnostic tools. I should say that this is clearly an R&D project, right? So we're still at an early development. We now have shown a proof of principle where we can isolate nucleosomes and purify them and do the mass spectrometry on them. But there's much more things to do, like looking at real tumor samples. But I think those results are very exciting, and I'm super excited to work with Volition on this particular topic. And one thing I would like